What's your preference?
Or you can knock on the door immediately to the right outside this room and I
Okay, we can make that happen. Okay.
do you have any questions?
Question, concerns, can you want to give my talk? That'd be perfect.
No, but for the academic progress of the participants, we should potentially reevaluate.
Let's just see here, because I'm terrible with technology because I don't see my PowerPoint right now.
So
And
you should be able to back here.
oh, but it's up there, so that's all that matters.
Yeah, you still though should be able to see it from here to continue to advance the slides on.
Yeah, so let's do this.
Perfect.
But I don't see it. So can I just like tile? How do I show all windows maybe? Of course.
It should be open just like behind this stuff. Drag it
But I
down.
think I'm like mirroring displays or something because I can't my mouse goes over here and then I lose it.
Oh,
So
you want to extend, not
yeah,
or extending, not mirroring. So if you go into your
my
preferences,
yeah, is that like up this here, this guy, open display preferences,
then you want to make sure that you are
arrangement. I don't want to
you wanna
mirror,
mirror, not extend. Yeah.
mirroring, we're mirroring.
Look at that. We're
Okay,
on the wall.
yes, okay, we are mirroring. Okay, all right,
And then for questions afterwards, we can just put the screen.
Yeah.
yeah. And I have no idea if people can see me, but
So as long as this is on,
yeah,
people will be able to see it.
okay,
You also will be able to speak into the room and they can speak back to the speaker.
okay,
The microphone on you right now is
Yeah.
on as long as you see this little screen.
okay, perfect. Hello, internet. Okay,
Okay, thanks. for
do you
that.
need this one or you can talk into this guy? I guess so.
Okay, good morning, ladies and gentlemen. Welcome to Respirology and Firestone research rounds. We had a few technical difficulties, but welcome to everyone who's here in person, and also we have a few people joining us virtually. Today's speaker is Jay Hirota, homegrown Bradford boy, who did his PhD here in the Firestone in animal models of allergic airway inflammation, supervised by Mark Inman. Jay then went off to UBC and spent a few years there, I think 6 years in total, working with the likes of Chris Carlson and Darren Knight and various others at UBC, expanding his horizons and the breadth of his research, which encompassed work that he's continued here on ABC for the protein. And more recently, he's sort of been crossing over a lot. He's been one of the leaders really driving the interdisciplinary collaboration between biomedical and the engineering sides of Master. And some of this is going to be the subject of his talk today. The title of which you can all see, Kaizen for preclinical models of lung disease, from static 2D to dynamic 3D and beyond. So very exciting stuff. I've seen some of this work already, and I would just like to say a special thank you to Jay for stepping into the breach at short notice to cover an open session that we had today. So Jay, looking forward to your talk very much.
All right. Thanks, Mark, for the introduction, and thanks tech support. Always helpful when everything works out. Yeah, when you said kind of stepped in on short notice, that's one of those engineering principles is you need to have constraints. If you don't have constraints, if you don't sort of define the terms of your problem and your solution, you'll never come to one because you'll just, I've got more time, I've got more money, I've got more resources. And engineers really work on constraints. And I can say that from firsthand because I'm married to one and she sometimes constrains me, but in the best possible ways. Best possible ways, I swear. Is this being recorded, right, Victoria? I'm in trouble. Definitely, this is important that we acknowledge the land that we are on, and we give thanks to Haudenosaunee and Anishinaabe nations, the traditional inhabitants of the lands where McMaster stands today, and where I speak from today. And we acknowledge a debt to those who were here before us, and that we should be grateful and look after the land that was theirs, and moving forward. And we consider the relationship that those peoples had with the land. Hopefully we can also have that same relationship with the land. Conflicts of interest, there is one with the provisional patent related to some of the work that we're talking about. So you can imagine I might spin it that it's the most impressive work we've done, but bear that in mind that there is a conflict of interest. I'd like to begin with a bit of the history. I've used this slide before. Some of you may have seen it. This is actually derived from my PhD supervisor, Dr. Mark Inman. And it's tried to be updated, and I may have missed all of the new faces that have joined Firestone in various capacities over the years. But really, there's a long history at the Firestone of innovation, innovation with and without commercialization, and I'll get to some of those. But we have really, from the very beginnings of the medical school and at McMaster, we have a lineage of leading clinical respirologists and researchers, but they also tinkered, and they also played, and they also built things. And I can probably do this, right? Is that acceptable? And so they also tinkered and they built things. And, along the way, they leveraged those things that they tinkered and built to then be at the forefront of clinical research on the global scale. And I believe Imran Satya has sort of said this thinking local but going global sort of mentality, and that was definitely laid down by the pioneers here at the Firestone on the left. Can you see my cursor? Yes. And actually, I can't get my laser pointer. But as we move forward, also, the group is growing, and there have been some changes, as in Paul has recently left us, as well as Shatil Ask and Martin Stampley have left. But that gives us opportunity to recruit new and fresh individuals that, okay, that screen's gone. And I've got some individuals here that have recently joined us, and I see Manali and Sarah here, and I'm putting the weight on your shoulders that this legacy needs to continue with both of you, and Terrence, as well as Kieran. And this is just a demonstration of some of those innovations that have happened, maybe not all here, as the Venturi valve in the top left corner that was developed by Dr. Moran Campbell. Moran Campbell being the founding chair of medicine here at McMaster, and as well being a respirologist. Sharing screen. All right, let's try this. This is going to be a real test of my abilities here. Share screen. We're going to do that. Share. I wonder why I didn't see this. Okay. Hopefully we can see this. Sorry for those virtually watching this presentation. But we also, Firestone, with the likes of Dr. Mike Newhouse and Mirna Dolovich, were involved in the development of the aero chamber, which, if any of you have had asthma or any of your friends have had asthma, there's probably been the use of the aero chamber with younger children, but even in individuals that are unable to use a metered dose inhaler. More recently, bronchial thermoplasty has really been pushed forward, and it's not working. Sorry, everybody. I thought I shared screen, but maybe I need to do. Okay. I could just do this, but if I go here, this is what happens. And it's maybe the presenter mode. Does anyone know that whole presenter mode thing? Slideshow. So if I go stop video. Okay. Stop share. Share screen. Desktop 1. Okay, so that might be it then. We're getting coached along here. And can we Can we get a amen? Amen.
It looks like you're sharing a screen. Is that what you're trying to do?
Yeah,
Okay, good.
we're hoping. I'm looking, it works now? Okay, apologies, everybody. I'm just, as much as we're trying to be innovative here, I am a little bit of a dinosaur when it comes to using these different platforms. But needless to say, there's been innovations in the past, and I don't want that to stop. And I don't think any of you should want it to stop. We should sort of hope for the care for the patients that come through the Firestone, but also globally to improve. And for that to happen, I think we need to think beyond just some of our basic research problems, but think about how we could translate those into the clinic and solutions for the patients and their families. Okay. And I have this Venn diagram where I ask the question, what will fuel the next generation of innovations here at the Firestone Institute? And I don't think there will be any argument that historically clinical care at the Firestone has really driven some of the research questions that we've had here, whether they're basic science questions or clinical questions. And we shouldn't lose sight of that. That's been very productive. It's been very helpful for the patients. We think about the various measures, whether it's induced sputum, whether it's 6-minute walk test, whether it's allergen inhalation challenges. A lot of these have been developed with the goal to really understand the challenges that the patients are facing and improve their quality of life. Along the way, there's definitely been an interaction with engineers. Engineers have helped bring those ideas to life. Sometimes you need someone that has the ability to build things, and engineers think differently. They don't necessarily think, well, we need to have a certain number of sample size to drive this forward. It's just, does it work? Yes, iterate. Does it work? Yes, iterate. Keep on going forward. And sometimes that, if we think about equity, diversity, and inclusion, having diversity of thought, having an engineer at the table, they think differently. Well, that doesn't mean they think wrong. It just means they think differently. And the injection of those different mindsets, I think, is something that we should continue doing, and we've done in the past quite well. I would also say that moving forward, we're not getting away from data. I don't want to just say bioinformatics. I think bioinformatics is really kind of restricted to the omics, whether it's genes and proteins and metabolomics. But data, I think, is not going away. And I think data could help sort of either identify problems, but also help inform some of those solutions. And I don't see us kind of moving away. This is my bias. This is my, hypothesis. I don't know how I'm going to test this, but I suppose maybe there'll be a research group that doesn't use data analytics and bioinformatics, and there'll be a research group that does, and we can kind of see, okay, in the next 10 years, who's bringing in the money? Because that's the metric that we're measured by. Anyway, just a little tongue in cheek, but I think we need to consider where data scientists and bioinformaticians can help complement the clinicians and the engineers. Today, the talk is an interdisciplinary talk, and I wouldn't be here without all of these people. There are some other students that I couldn't get their mug shots in time, but this work wouldn't be pushed forward without a variety of people. The faculty that deserve note are, Ravi really welcomed me with open arms when I first came back to McMaster. He is now a university professor title, distinguished professor, mechanical engineering, and always a very dynamic mind. Bo Yang is a recent recruit in the last 5 years. And I think all of you should be aware of his work, incredible work that he's doing in engineering new models and advanced models and tissue engineering. Jose is a wonderful individual who's the director of the Center for Advanced Light Microscopy, a very, very broad and interdisciplinary individual who understands microscopy, understands materials, understands chemistry, and the links between them all. Scott is actually an educator in engineering. He's an award-winning educator in engineering at the University of Toronto. And he was the supervisor of a very important person, Crystal, with the cat ears down in the bottom right. And Yaron, our friend in thoracic surgery just here, has also been crucial for providing us tissues that we needed for some of the work that we're doing, ethically approved sourced tissues. And the students here, I mean, in the audience, we have Jenny here, and we have Ryan here from my group, Kathy here, just making sure I'm not missing anyone. But those are the individuals. Laura, you're in the back there. The mask is there. That's my excuse. And the work really wouldn't be pushed forward without the students. I'm very happy to have them as part of the team. This is just a bit of a plug that if there are people within the Firestone or even in the broader Hamilton community that need access to 3D printers for rapid prototyping of things, if you want to test something out that might be helpful in the lab, we have the capacity here. These are just the 3 printers that are available, and we may be expanding this capacity. They have different build plates, so sizes of things they can build, different resolutions, and you can really work up some ideas quickly to see if they work, and if they don't work, you can iterate from those and change and make improvements. And this is really that introduction of that engineering mindset within the group. We also have bioprinters that aren't just printing resin and plastic, but they are actually printing materials that can support cell growth, cell proliferation. So essentially printing some tissues, rudimentary tissues. There's 2 systems here. The Allevi 3 is in Jose's lab on campus, and the Aspect Biosystems system is up in our lab on the 3rd floor here. And we'll show you some data that's been derived from these 2 systems. So with that little bit of an introduction and pitch that I think engineering is important, and merging it with the traditional biomedical research could provide new solutions. I want to talk about some of those new solutions that we've been trying to push forward in the lab. And being a basic scientist really does involve us playing around with cells, maybe some animals, but staying in that lane. And that lane has traditionally been occupied by hard plastic dishes, 2-dimensional models. They're static. There isn't any airflow or stretch going on, just like our lungs right now as we're breathing. That's what's happening. Well, why are we modeling it on hard plastic in the lab that's static? And so I was just asking myself that question. And I said, well, why don't we try building models that better represent the human lung? And I use this Picasso's The Bull because there's multiple versions of a bull. There's multiple models of the bull. And I don't think anything actually recapitulates the real bull, but can we get something closer to what that bull is? And in our case, closer to what the lung is. And so I'll begin by describing a model that introduces flow, and in our case, airflow, into our in vitro systems. So as Mark mentioned, I spent some time out west, and I was involved with Chris Carlston, who does a lot of air pollution exposure work, whether it's like diesel exhaust, or whether it's particulate matter, or even wood fires, wildfires. And to model those exposures in vitro, there are a variety of ways to do it. And if you think about it as a spectrum, there's really approachable, almost reductionist models on the left, which have conditioned media exposures. And then you have these really dedicated pieces of infrastructure that are complex. They measure everything, but they might be a little bit more than you actually need to ask your research question. In the middle, adding to that mix of the decision-making process is, well, what cells are going to use? Is it going to be, primary cells or a cell line? Are you going to grow them on 2D or potentially grow them on a transwell to make them sort of 2.5 dimensional? And if we're going to expose them to smoke, how are we going to expose them to smoke or air pollution? And so, we were looking at this and we said, well, we definitely don't have the money or the space to get one of these complex exposure systems. But every time we did something on the left with the conditioned media, which is where you just bubble smoke through your cell culture media, the reviewers and/or grant reviewers would say, well, what is this really a model of? And so we were kind of stuck. And that was the problem. The problem was, how can we actually deliver smoke or combusted material to our cells to appease grant reviewers, but also to make a more physiologically relevant model, which is smoke exposure to the lungs? And so with the help of Mode, who's not here, I think he's writing a review right now, we developed the idea was make something that is printable with our 3D printers, disposable, it's cheap, and it's very easy to use by those labs that aren't necessarily dedicated for air pollution exposure. And what we have here, hopefully, yes, you can see my cursor, it's just like a manifold where we can control the air coming in, whether that's fresh air or smoke. And then within each of these 4 chambers that we have, we can place a transwall insert. This transwall insert is commercially available. It's standard size. So if you go to Corning and you buy a transwall insert, it will fit into this system. So the idea was that other labs could buy off-the-shelf product, combine it with this open source print, and now begin doing exposure work. In our application where we were looking at, we were trying to wonder, well, what are the impacts of cannabis smoke exposure on airway epithelial cell function and viability? And so in our case, we're combusting cannabis and we use a syringe to pull, acting like an inhalation, and we generate that smoke. We have a little valve that we just spin, and the smoke funnels through the tubing onto the cells. And then if we're going to model smoking, people don't smoke all the time. They smoke, they inhale, and then they have fresh air. So then we can cycle that through with whatever you want to control. If you want it to be 3 times the amount of fresh air, 4 times, whatever it happens to be, we can introduce fresh air to the system. With the engineers, they're able, just like the bioinformaticians and the data scientists can sometimes throw up heat maps and little connecting diagrams where they show all these wonderful relationships, and there's probably only 1% of the people in the room know how those data are generated. Similarly, engineers can do that. In this particular case, this is a fluid dynamic modeling. We wanted to make sure that each of those chambers was actually getting hit with the smoke. It was a good idea, but if they're not getting the smoke, and if it's not equal distribution in each of those chambers, then we're going to get variability within that plate. And the take-home message here, hopefully you can appreciate, is that in the center of the wells, these are the 4 chambers, there's the same amount of blue. And the same amount of blue means the same amount of velocity of air that's getting into that chamber. And we're using carbon dioxide as a model gas. And so we just model the flow of carbon dioxide within the chamber, and we show that that distribution is equal. So that was reassuring for us, at least theoretically, that the distribution of the gases in each of those chambers was equal. So that if we had smoke, then each of them were going to get hit similarly. But another concern for us was, could we actually empty the chamber? Or if we put smoke in, would that smoke stay in there? Would it settle in there? And would that really then compound and stack on top of each other so that these cells were just always getting smoke and never getting any fresh air? This simulation, again, it's a math simulation where the color coding hopefully is intuitive, where we start an inhalation here on the top, and again, carbon dioxide being the model gas, and through 5 seconds of inhalation in one of those chambers, working on the assumption that all of the chambers are the same now, we can get filling of that chamber with the carbon dioxide. And then following that 5 seconds, we start an exhalation protocol. So just now we've taken our smoke in, now we're going to exhale. And in our case, we're going up to 20 seconds. And we see that at the bottom of that chamber, we do get that carbon dioxide going in there. So there is carbon dioxide, or there is a gas, if this is cannabis being combusted, that is getting to the cells, which are residing here at the bottom. But then through the next 15 seconds, when we're introducing that fresh air, the chamber gets flushed and we bring it actually back down to the starting point. So we're not getting stacking, we're introducing fresh air. And now at this point, if the experiment is designed that way, we could introduce another puff of smoke or another puff of wood burning or biomass or anything else that you're using. Now, those were theoretical models. That was more of the engineering side. But then marrying the engineering with the biomedical research, well, we need to make sure there's like a functional response that is indicative that those cells are actually getting hit with what we think they're getting hit with. And in this data, this is actually primary human airway epithelial cells grown on transwells and getting hit with that cannabis combustion, the cannabis smoke. And then what we did, we took the cells off and we looked for gene expression. And so we did RNA sequencing on it. We wanted to see, what was up, what was down. And these 2 genes that are up, these are involved in an oxidative stress response. We don't need to go into all the biology of them, but essentially, if there's going to be either pollution, whether it's cigarette smoke, whether it's tobacco smoke, these genes will go up. And so it's a bit of a positive control to say, okay, well, now we know those cells are getting hit. They're responding as they should. And whether that's as they should, as if you take human samples of individuals that have been exposed to pollution, they'll have an upregulation in these genes. So it was giving us confidence that we were accurately modeling what would be happening in the human airways. I throw in this other gene, TXNIP, because in the paper, what happened was when you use that submerged monolayer system, TNXIP went down. But in this air-liquid interface system with real smoke, it doesn't change. And so the part of that was saying that, okay, not all models are made equal. And depending on what your genes you're interested in, the process you're interested in, you might need to pick one model over the other. And for us, we think whole smoke exposure is going to be a more accurate representation rather than smoke conditioned media. Now we've been taking this model a little bit further, and this is unpublished data, because we wanted to look at how cannabis smoke impacts immunity. It's a bit of a gap in the field because of the legal issues around cannabis. And all of the work that had been done with tobacco suggests that tobacco smoking impairs immunity and leads to greater susceptibility to influenza infection. The corresponding data with cannabis smoke doesn't exist. You could make the hypothesis, which we do, is that cannabis smoke is going to attenuate antiviral immunity to influenza. And that's what this experiment is trying to get after. We grow the human airway epithelial cells, we grow them at air-liquid interface, we give them a smoke or air exposure, and we let those cells go overnight. And then the next day, we hit those cells with an human influenza virus, and we let that go overnight. And then in this case, it's a single readout, which you're going to see, which is CXCL10, which is, it's a good marker because it's known to go up with influenza. There's actually quite a large signal that it should go up with influenza. So as a positive control, if we don't see this going up, we know our model's not working or it's not representing what's known. And it would be a good readout to see the impact of cannabis smoke. And that's the data that we're presenting here. And again, mindful that it is unpublished data and it is preliminary, with an N of 5 in some, or 4. It's 4 actually here. And what we can see is that cannabis smoke alone isn't really impacting this CXCL10, whether in the apical side, the top side of those airway epithelial cells, which is exposed to air, or the bottom side, the media that's feeding those cells. Cannabis smoke alone doesn't seem to be doing anything. But when we infect those cells with H1N1, we do see an increase in the CXCL10 on the apical as well as the basal compartments. Not statistically significant, but typically with primary cells, we have to get up to about 8 or 10 due to the variability. But then what we do see is that if we have pre-exposed those same cells from the same donors to cannabis smoke, then hit them with the influenza, they don't seem to be mounting a CXCL10 response. So this just as one readout, it suggests that there's a dampening of an antiviral immune response with cannabis exposure. And so that's ongoing work in the lab using that 3D printed exposure system. Okay. I'm seeing my time, 18 minutes. Okay. So again, trying to advance beyond just using our static systems on submerged monolayer, we also recognize, yes, there's airflow when we breathe, but there's also some stretch in the lungs that happens. And again, your static plastic systems don't incorporate that. So how can we do that? And actually, is that even important? And this data is from Martin Kolb's group years ago now, almost 7 years ago now. And they demonstrated in this paper, and I'll get to explaining the data, how important stretch is in modeling disease processes and what we might be missing in a static system. This is just a demonstration of what we can get through our collaborators, including Yaron just down the hall. We can get some lung samples. We can get lung from non-IPF or healthy individuals. We can also get material from IPF patients. And in this video, hopefully you can see, and I know it might lag for those of you watching virtually, but this apparatus up in the Kolb lab does it. Oh, you can actually hear it too. There's audio. That's fascinating. All right. Got to go back. Is that audio from me or is that audio somewhere else? Okay. And so what happens when you actually stretch this? This is a system that the Kolb lab has upstairs, where you can stretch this tissue and do the type of stretch that would be similar to just resting breathing. It's not a cough, it's not a massive movement, but just resting breathing. Does anyone else hear that? Is it coming from here or is it coming from outside? Okay, so it's not, I don't think I have that sound in here. So sorry, virtual people, but there's some noise going on. But then when you actually take this lung tissue, and this is before stretch, okay, with non-IPF, or before stretch with IPF, and this is active TGF-beta. This is measure, one readout of a fibrotic process. Well, in the non-IPF lung, and hopefully that's a variety of us and all of us in this room, there's really not this active or this intrinsic TGF-beta pathway being active. But in the IPF patient, unfortunately, there's a baseline activity of TGF-beta. When you stretch it, it's almost double. And so if you think about what we're modeling in the lab and the static system, we're missing 50% of that signal. And maybe that's the signal that we can go after. Maybe there's a unique mechanism that's governing that active TGF-beta during stretch. And so this is the argument that the Kolb lab has definitely been pushing. But it mirrors some other work in the epithelium that if you don't model stretch or introduce stretch, you're missing part of the equation. Mucus secretion is also influenced by stretch. And so if you're looking at mechanisms governing mucus secretion, which are relevant in a variety of chronic respiratory diseases, what are you missing in your static system? And don't worry, I'm missing those same things on a lot of things we do, but it's the questions that we're asking ourselves. Now, this is where I'm not on social media anymore, and this is one of the things that you miss out when you're not on social media. During COVID, Crystal here was tasked with still teaching a engineering graduate level course and being the TA for this course. And Crystal, speaking of constraints, was willing to take that constraint head on. And she created, using 3D printing, a lot of these plastics are 3D printed, using her connections overseas. And she was able to create this do-it-yourself mechanical tester for her mechanical engineering course that she, assembled and put into boxes that she then shipped out to all of her students. And the students had responsibilities to put it together, but then test some of the materials. They were handed out with the kit as part of their lab course while doing this all through COVID in the safety of their own home. Incredible. And Crystal tweeted about it. And I said, what's going on here? Like, this is incredible. And so I actually reached out to Crystal and I said, we really want to introduce stretch into biomedical systems and lab experiments. Can you help us? Can we? And she again loved the challenge, and it's been a very productive relationship. And I'll get into how we took that device, we iterated on it, and we evolved it to be a cell and tissue stretcher for our group, which we are calling the CAT stretcher. This is all do-it-yourself. This is all open source. There's nothing proprietary here. And it includes a lot of off-the-shelf components, including Raspberry Pi, Arduino controllers. Now, there is a custom PCB with a printed circuit board, but the design plans for that and how to get that created or manufactured are available online. And the rail system, as well as the motor to drive it, also all accessible. This comes in probably about $500 to make this system versus $25,000 for commercially available systems that are not modular. You can't, they're not open source. You can't change them as you go. And so that was from Crystal's sort of a version 1.0 to what we were looking for, version 2.0 in the lab. This is what we have now called the cell and tissue stretcher. It uses a very easy to use graphic user interface which eliminates the need to be an engineer or a computer scientist to program anything. It's literally like, I can do it. You can just kind of, okay, I want to ramp up. I want to increase the time for my wavelength. I want to increase the time that I stretch for, the frequency that I stretch for. All of that can be programmed just with this very easy user interface. These are the, would be your experience if you're using the system, where you can change the waveform. You can change, if it's all color coded. So if you want to change, okay, well, this is an inhalation. Let's change the time for that inhalation. And let's have the individual hold that inhalation for an extended period. You can tailor that to what you're looking for. Or you can have a breath pause down here. All of these you can change, and as well as the runtime. We've ran it, I hope I'm right, but I know we've ran it for at least 7 days, but I'm not sure if we've gone even further than that. I'm looking at Ryan, but I see a nod. Okay, so we've gone up to 7 days. Really, it's not an issue of the machine. It's an issue of the media on the cells, and the cells, whether we need to top up media on the cells with them drying out. But the machine is able to run continuously. We made it so that you could program in your preferences. So if you had a particular experiment that you were doing, you could, load that in, lock it down, and then every time you came back to the cat stretcher, you could run your experiment as needed. The one component that we wanted to do was make sure that it was modular. Those commercially available stretching devices, they typically can only do one thing, whether it's the Martin Kolb's group where he's stretching tissue. Well, that's not amenable to submerged monolayer cultures. But then the other systems are only for submerged monolayer cultures. Well, what happens if we have live tissue, or what happens if we have cells at the air-liquid interface? Can we make a system that accommodates all of those type of experiments? And indeed, the cat stretcher is able to do that through these chips that we design in our lab. This is your traditional submerged monolayer, which would be your culture dish, and the cells would grow on the bottom here, and we would just simply stretch that back and forth. I'll get to some videos later on. This is a organ-on-chip device where there's fluid flowing through the bottom, and we could either have fluid on the top if we like, or we can have air on the top, or smoke on the top, or another exposure on the top, and cells will grow on a membrane within the middle of that chip. The other middle chip design is a hydrogel chip. Hydrogel, think of it as it's a material that can absorb a lot of water, but still have some structure to it, and maybe it can be spongy or stretchy. And we can make these hydrogels with collagen or long extracellular matrix, and then make a 3D type of material that we can then stretch in these chambers. That's the middle one here. And then the last being, okay, well, like the Kolb lab, they have the tissue stretching. Could we also accommodate tissue stretching on our system? And this is a magnet clamp that allows for us to clamp down on a piece of tissue and stretch back and forth. And this is what each of those chips looks like when they're on the cat stretcher. There's these metal poles, we can slide those on, and hopefully you can appreciate that there's room for multiple chips to be on. So we have medium throughput, let's call it. We could stack chips if we need to. So unlike the apparatus in the Kolb group, which is great, it has, it's very sensitive, but you can only do one tissue at a time. And so you're introducing an incredible amount of variability when you do one experiment one day, the next day, another experiment, the next day, another experiment. We can do all those experiments all at the same time and hopefully tighten up the data, run controls as well as experimental groups at the same time. Now, this is a video of how these chambers go on, because I know I sort of said there's these pegs and there's these different chamber sizes and formats. The idea was it was all modular. It uses the same principle to attach. So these stainless steel pegs, it does help if you do a little bit of a lubrication, I believe, to slide them on. Otherwise, the chambers can be a little bit sticky and not want to slide on. And this is a demonstration. The square ones are the submerged monolayer chambers. In the more rectangular chip designs where there's multiple chips on or channels on the same chip, Those are the hydrogels. And here you should see just a demonstration of the speed and the stretch. And so you can imagine cells growing on that submerged monolayer, experiencing that stretch right now, going back and forth. You can do it acutely. You want to do it for a minute, okay, or do you want to do it for a week? And that's at your discretion. As described, the stretch is completely up to your discretion, and the speed is up to your discretion. And we're even going as far as trying to model cough, so a very accelerated movement, one quick movement, and then okay, now we'll go back to normal breathing, one quick movement, back to normal breathing. And can we model cough? And we're trying to do that with IMRAN. And this is just removal of the chips from the system. And the last one that I wanted to show you is the actual tissue chamber. Hopefully it comes on. Okay, so this is the tissue clamp system, where you can toggle the cat stretch to move it back and forth. You just toggle it to the length that you need. In our case, this tissue clamp system is a little bit of a longer chamber. Doesn't have to be, but that's our design. And we can slide that on, and you'll see these clamps coming on. They just have very, I don't want to call them teeth, because that would sound like it's going to damage the tissue, but there's little fingers that the tissue will get clamped in between with magnets. And then we have a region in the middle that actually is going to experience the stretch. In this case, it's just an artificial tissue in this image or video. The clamp tissue we try to avoid using. You can understand if we clamp down on a tissue, we might cause some damage or some cell death. So what we'll do is we'll clamp, and then once we do our stretching, we'll cut out the middle piece of the material. That's the piece of the material that experienced the stretch. It didn't get clamped. This is what we're going to use for our downstream analyses to determine the impact of stretch on that cell or tissue type. We can just fill this with media, and I believe this will start. Hopefully you'll see it move slightly. Okay. And then there will be a little bit of a video of us doing live lung tissue in a hood. Live lung tissue being stretched here. So this is coming from your on, and we thinly slice live lung tissue. We get it to about 3 millimeters in length, and then we put it into these clamps, and then we stretch it over a defined period of time. Okay. Now that's all great. That's a wonderful tool, Jay, that you can stretch things. What is it actually doing? Is it actually recapitulating? And this is coming back to the biomedical side of things. Okay, you can engineer that, but is it actually recapitulating what is supposed to happen with your material or your lung tissue? And one of the things that happens with introduction of stretch with fibroblasts can be the transformation of a fibroblast to a myofibroblast phenotype. And that transformation from a fibroblast to a myofibroblast is accompanied by an increase in alpha smooth muscle actin. And in our case here, we had, I think there were 9 actual experiments. These are just representative blots, and it's variable and it's donor dependent. But what we have, these are just 3 that are controls that are non-stretched, 3 that are stretched. This is the alpha smooth muscle actin that we're quantifying. This is a total protein blot. This is how we normalize blots in the lab. And when you do that normalization with all of the experiments that we completed, we see an increase in the alpha smooth muscle actin expression in the stretched tissue versus the control tissue. And in the top here, this is what we would suggest if you go down and you want to work with us and use the system, is you should treat your controls the exact same way. You should cut them the exact same way. You should put them in the chambers the exact same way and have them in the same environment in the exact same way. They just don't get stretched, because in that way, then we can, any handling that might influence the readout is being mirrored in your controls relative to your treated groups. Okay, now I want to close off here with how we're moving a little bit beyond flow and stretch, but now how do we introduce 2D biochemical or physical environments as well as 3D environments into our model systems? And I'm highlighting here human lung extracellular matrix. I come back to that hard plastic and I say, well, how similar is that hard plastic to our lungs and the environment that our cells in our lung are growing in? Probably not. So can we introduce some of that human lung extracellular matrix into those hard plastics, or maybe even into 3D model systems? That's the question we were asking ourselves. This is a protocol that we've worked up in the lab. It is based off a considerable body of literature, but we needed to optimize it for lung tissue. Happy to share the protocol and work with you. Laura, who is here, has been driving a lot of that. I think even some decellularization may be going on right now for some tissue yesterday. But ultimately, we get this tissue and we need to cut it up into tiny little pieces. So the detergents that we treat those chunks of tissue with are allowed to break up the cells that are present in the tissue. And they break up the cells and we wash them around and we rotate them. And what comes out of it is a final product that doesn't, and I'll show some of the macro views of it, but it's the final product that the cells are all gone, but the matrix remains. The framework of the lung still remains. Now you could use that right away. The Kolb lab has used that decellularized framework and repopulated it with cells. Other groups are doing tissue engineering where they take a scaffold of a human lung and they repopulate it with a donor's lung or donor lung cell to try to make a transplant. So you could do that, or we could go even further. We can lyophilize it. We can grind it down into a powder. And now we have a powdered human lung that we can use to coat surfaces or introduce into our models to introduce that extracellular matrix that is actually in a live human lung. This is what it actually looks like when you start. You can have these pink, fresh lungs, chop them up in tiny pieces. And then after a series of washes over a series of days, the pinkness goes away, the cells are all gone, and you're left just with a shell of the formal lung that contains the extracellular matrix. And there's that protocol, that schematic I was showing you. And if you sort of take a piece off at this point where it's decellularized, here's what the histology looks like. There's your control that wasn't decellularized. This is just your H&E. The blue nuclei are present in your control tissue, but in the tissue that has been decellularized, hopefully you can appreciate no nuclei here. So all the cells are gone, but what remains, you can see it there, is the scaffold. We've quantified it in the paper. We show there's collagen there, there's elastin there, there's some glycosaminoglycans there. There's other ECM components still there. So we can use that to influence cell behavior in our models. We can make gels. I sort of talked about this hydrogel. We can make hydrogels out of this material. And again, it's sort of like a jello-ish type of material. It's actually not unlike jello. And within that gel material, you can see these fibers form, these extracellular matrix fibers. There's pores within those fibers. We're working to fine tune the size of those pores and the strength of these fibers and the size of these fibers. Now, if you go further, I told you we can make this powder and maybe we can just coat hard surfaces. They're still hard, they're still static, but now we've introduced some biochemical cues that might be present in the human lung. And we've done that here where we've taken fibroblasts from a non-IPF person, we've plated them down on ECM from a non-IPF person, or we've plated them on ECM from an IPF person. And hopefully you can appreciate that there's a little bit more green in the fibroblasts that are grown on the IPF ECM. There seems to be this intrinsic push from the IPF ECM to push these cells to overexpress alpha smooth muscle actin, maybe become a myofibroblast phenotype. So this feed forward mechanism with these cells driven by the matrix. And we can also do, this is just from a grant application, so that's why this figure's here, but you can imagine, okay, you grow those cells on different matrix. Well, I'm not interested in protein expression, but I'm interested in gene expression patterns. Well, you can isolate the cells and do detailed transcriptomic work on that as well. And we see differential gene expression patterns when the same cells are cultured on the different ECM. So the ECM itself is giving cues to those cells to behave a certain way. Now, the second ingredient in these advanced models is this concept of bioprinting. I showed and very early on some of the infrastructure that the group has access to, but I didn't really expand on what bioprinting is. There's a variety of ways to bioprint. One of the simplest ways, most common ways, is this extrusion method. Think of it as a syringe. There's a tiny needle, and what comes out at the end is actually the bio ink that contains some cells, maybe contains some factors that support cell viability. And that material that's in the syringe, we call it a bio ink. And through some interesting chemistry, whether it's through light or whether it's through an enzymatic reaction, we can lead to cross-linking. So we turn that ink, which is fluid, and we can turn it into a gel-like material. We cross-link it on the fly or relatively quickly and turn it into a material. So ink here on the left, but when we cross-link it, we can cross-link it into, well, there are limits on the resolution, but we can cross-link it into whatever shape we want. In this case, this is a grid, and each of those holes is 3 millimeters together. So that's ingredient 2. We've got the ECM, one ingredient, bioprinting, another ingredient. And for those of you that haven't seen any bioprinting, this is a model that sort of fails. This is a bio ink that fails. It doesn't have the material properties that you need. It's not stiff enough. It doesn't gel quick enough. And it's supposed to be a nice tube. It's supposed to be just a cylinder, a hollow cylinder, but it's sort of falling on itself. It can't support its own weight. This is sort of a test in the bioprinting space for your inks. Your inks need to sort of pass this test, and if they don't, then there's limited applicability, at least in the conventional sense. But our very talented Moran Mirabel lab has come up with a solution. Now, hopefully you can see that there's a little bit of bluish light that's going on here. And this bluish light is activating a UV, or let's just call it a light activated cross-linking agent. And so the light comes in. I think people, I honestly don't do my nails, but I think this happens similarly with people get their nails done. It's a gel, and then you just put them underneath, and then it hardens. Same principle here. Same principle is happening. And so we're using this light to activate, and now you can see that this is not collapsing on itself. It's passing this test of supporting its own weight. And this is sort of the basis of that provisional patent application that Mabel has pushed forward. I'm mindful of the time here, so I'm just going to buzz through. We can tune these materials so they are flexible. Okay, you can stretch this. You'll see that kind of looks like human lung or any other human tissue. It's not hard. These are just forceps that are naturally opening. We're not pushing them open. They actually just naturally open. And depending on how stiff we want it, we can tune it to be that stiff. So a fibrotic lung is going to be stiffer than a non-fibrotic lung. So can we tune that? Indeed, we can. We can operate within those ranges. These are just measures of stiffness, where we can have a non-IPF lung or an IPF lung, and our bio ink is able to be tuned to support those different properties that are needed. These are the resolutions. Depending on how we tune it, there are trade-offs. If you make it too loose and too, stretchy, then it doesn't actually harden quick enough, and you lose a little bit of the resolution. And so there are trade-offs with this. It's not like you can print everything within your head. There's constraints you need to work with. Importantly, with this particular ink, we can introduce that ECM. So that's one of the first ingredient. We can introduce the ECM into this bio ink now. So we can combine ingredient 1, the ECM, with ingredient 2, which is the bio ink. We can also add in fluorescent labels as needed. And then we can add ingredient 3, which is the stretching with the dynamic forces. So if we add all these ingredients together, the goal, hopefully, in this final recipe, this food analogy, is that we can have a tunable bio ink that we can make 3D models that can support cell viability, and they're amenable to stretching, and they have the same mechanical properties as human lung in health and disease. And this is that tissue clamp system that we've been describing. This is the demonstration, the green little dots that may or may not show up. This is a demonstration of cells being alive after we've printed them and after we've stretched them. And so with that, we are definitely focused on the lung space and modeling healthy lung, but also fibrotic lung, or a lung that has a stiffer mechanical properties. but the technology can actually be pushed in a variety of ways depending on what your research question is. And so with that, hopefully I've kind of just peppered in some published data, but also unpublished data on how we're trying to push forward our models. And we're not critiquing other people, we're critiquing our own models that we've always been doing. And we're just trying to push it forward so that we don't get bored, or at least I don't get bored. So with that, thanks for everybody. Thanks for the attention. And obviously thanks for the support for the all the research.
Great. Jay, thank you so much. Really, really wonderful talk. I mean, I think it's certainly for the older crowd amongst us, it's for me personally, takes me way outside of my comfort zone, which I think is a great thing, and it stretches our imagination as well as stretching the tissue. So, do we have any questions? If you're on line on Zoom, please put some questions into the chat, which I right now cannot see. Do we have any questions from the people in the audience here and now?
I'm very mindful that I went through that relatively quickly. I was trying to hit timelines, hit constraints.
So I have a few questions myself. The first thing that occurs to me is that it would be so wonderful, looking at your stretchy chambers where you have lines going in and out, meaning you can perfuse, right? So, what's the art of the possible in terms of not just perfusion, but I'm thinking of like real-time, continuous or semi-continuous measurement of factors. I mean, that it's quite dilute, it requires a lot of sensitivity, but we know that people like Leila Suleimani are making these devices that can measure things in real time as opposed to just cross-sectional.
Yeah, I think so. The organ on chip sort of technology where you're having something come in, but also something come out. This constant perfusion. It's really the art of possible is there. It's just if the sensors are there, whether it's Leila or Yingfu Li or even commercially available sensors. You could imagine a microelectrode that's measuring pH. There's the ability to just do real-time pH measurements in the effluent. And you could, since it's being perfused, maybe you don't need to introduce buffers into your media, which would buffer any pH changes. And then you would have online or inline measurements of pH in a non-buffered system. That's entirely feasible, depending on what I use pH because you can get off-the-shelf pH electrodes, but if it's something like ATP or if it's something like a CXCL10, then you may need a different means to measure it.
Yeah. Okay. Question. People must have questions.
Just everyone thinks that this guy is out to lunch.
Sarah.
So I'll repeat the question there for the audience. This was Sarah Svenningson, Dr. Sarah Svenningson, asking about what the 3D resin printers could print with and for applications in her group, but I think this is relevant to others. There are the resins that are available, different quality resins. So Form Labs will be able to describe the resins that they have available. There are resins and they're different quality resins, and they have different properties. Some are flexible, actually. Some of them aren't. Some of them can be autoclavable. Some of them can't. I'm not quite sure at the porosity of all of them, but presumably the properties are going to change as if something's stretchy or versus something that needs to be autoclaved. I know that's sort of one of their business models is to just sell the printer, but actually they're selling the inks or the materials. So presumably they're always introducing offerings. So I would advise you to just see what's available. And, if you need something that's autoclavable that has this sort of resolution ability, then we can give it a shot. So the build plates are constrained. There's that word. So, you know, I don't know what I say, like a bill plate of 6 inches by 6 inches. I think, you know, probably height, maybe you get up to that. But then as an engineer, you would probably just find a solution to work around that, build it in multiple pieces and assemble it. So if your build actually needed to be this big, okay. But alternatively, there are other bigger printers that are available that locally that we could just, you know, contract out as needed for any builds.
Jay, I also have a question about stretch in the sense of bronchoconstriction. So in asthma, you know, listening to what you're saying about alpha smooth muscle actin, I was reminded of Margaret Kelly's work when she was here, basically showing that, you know, when you do an inhaled allergen challenge in an asthmatic, of course, you induce bronchoconstriction. And when you look at the biopsies from those tissues, you see this transformation, well, of something into alpha smooth muscle actin positive cells. So fibroblasts into myofibroblasts, or maybe the smooth muscle into myofibroblasts. Presumably, there are opportunities here to incorporate smooth muscle, for example, and then just to use things like acetylcholine, etc., in order to study that transformation.
Yeah, that's a really good point. I remember that paper, and it was kind of controversial because it was in 24 hours, there was an increase in alpha smooth muscle actin in the submucosa. We've been really focused on stretch rather than sort of there's going to be different mechanical forces in stretch than there would be on contraction, squishing. So that's just something for me to think about. I don't want to.
I mean, if you stimulate the muscle with acetylcholine, for example, it will contract, right? And if you could put it under some kind of tension, so it's trying to contract under physiological tension.
And is that is that sort of mechanical transduction happening?
That's switch on G.
Yeah, I mean, again, we could put it under stretch, and in the bath system, introduce acetylcholine, and obviously have the corresponding controls, and then you sort of see like, okay, well, how much stress does it need to be under for that transformation to happen? And it could be done. I was thinking, so yeah, absolutely. Okay.
Okay, any last minute questions?